rapid access to comprehensive oncology data and survival analysis. This is the OncoToolkit.
Risk stratifying a new multiple myeloma patient, it takes up critical time in the clinic. You're flipping through guidelines, calculating scores, and let's be honest, this isn't just inefficient, it's a potential clinical risk. What we need are instant evidence-based answers.
And the core of the challenge is right here. Myeloma is just an incredibly heterogeneous disease. Some patients will have a really slow-moving, indolent course, while others have extremely aggressive disease right from the start. That's why getting risk stratification right isn't just some academic exercise, it's absolutely essential for deciding who needs more aggressive therapy or maybe needs to be on a clinical trial.
So the goal here is pretty simple. Let's transform that complex, manual process into a single, streamlined calculation. Give you back your time, but without sacrificing any accuracy.
Okay, so let's get into the science of it. Because to really understand how a tool like this works, you got to appreciate the clinical evolution it's built on.
The whole journey of risk stratification has really been about getting more and more precise. It all started back in 2005 with the ISS, which just looked at tumor burden. Then, a decade later, the R2ISS brought in disease biology. And now, just recently, we have the R2ISS, which gives us this even more granular additive score. Let's break each of these down.
So the original International Staging System, or ISS, was really the foundation for everything. Its real power was its simplicity. It just used two blood markers that are available everywhere, beta-2 microglobulin and albumin.
And here are the specifics. For stage 1, you needed a beta-2 microglobulin under 3.5 and an albumin of 3.5 or higher. Stage 3 was defined by a beta-2 that was 5.5 or more, and stage 2, well, that was basically everybody else in between. It was simple, it was effective, and for its time, it was a huge leap forward.
But, we all knew that tumor burden wasn't the entire story. So the revised ISS, the R2ISS, was the critical next step. It started to integrate the markers of aggressive disease biology, things like elevated LDH, which points to rapid cell turnover, and those high-risk cytogenetics from the cancer cells themselves.
Now here's what's so smart about this. The RIS doesn't throw out the old system. It builds right on top of it. So to be stage 1 now, you need that original ISS stage 1 status, plus a normal LDH and standard risk genetics. And to be stage 3, you need to be ISS stage 3, plus you need either a high LDH or one of those key high-risk genetic markers, like deletion 17p or the T4/14 translocation.
And this just brilliantly illustrates why that revision was so powerful. These survival curves show a dramatic, statistically significant separation between RISS stages I, II, and III. This isn't just some theoretical model, right? This has real, proven prognostic power in the clinic.
Which brings us to the latest evolution, the second revision, or R2ISS. And the whole goal here was to add even more granularity. The problem was that the RISS stage 2 group was huge and had a massive range of outcomes. So the European Myeloma Network came up with a points-based system to really tease apart the risk within that group.
And here's how it works. You can see each risk factor is assigned a specific point value. So ISS stage 3 gets you 1.5 points. Things like high LDH or a deletion 17p are worth 1 point each. And a newer marker, 1Q gain, that's worth half a point. You just add them all up, and that final score places a patient into one of 4 really distinct risk groups.
So, we've just covered all these complex rules, the cutoffs, the different scoring systems that you'd normally have to go look up manually. Now, let's see how all of that clinical science gets packed into one powerful automated solution.
So here it is, the app's interface. And you'll recognize these inputs immediately, right? Beta-2 microglobulin, albumin, LDH level, and the key chromosomal abnormalities. These are the exact standard of care data points we just went over, all laid out in one clean interface.
You just plug in the data, hit Calculate Score, and bam, the result is instantaneous. No more looking up tables, no more doing math in your head. You get the precise RISS stage and the 5-year overall survival estimate that goes with it, pulled right from the primary literature. That's something you can use for clinical decisions and patient counseling right away.
Okay, but for any clinical tool, especially one that automates complex guidelines, trust is everything. Speed is great, but transparency, that's completely non-negotiable.
And that really brings us to our core philosophy here. We just don't believe in black boxes. As an expert user, you have to be able to verify the logic. So the math, the reference tables, it's all right there for you to review.
And this is the most crucial part. With one click, the app literally shows its work. You can see the exact formula logic it's applying, and right next to it, the source of truth data table pulled directly from the original publication. You can trace your inputs all the way through the logic to the final output. It's about complete transparency.
But the value here doesn't just stop with an individual patient. This tool was also designed from the ground up to be a powerful bridge from what you do in the clinic to your academic work.
So after every calculation, you can save the result, maybe with an optional patient ID, and over time, you start to build this cohort of stratified patients. Then whenever you're ready, you can export that entire dataset, all the inputs, all the outputs, into a clean, research-ready CSV file with just a single click.
And that export is just the first step in a whole research ecosystem. You can take that raw data, use our free CSV Polisher tool to instantly clean it up and handle any missing values, and then upload it directly into our survival analysis tool.
The entire workflow is designed to be completely seamless. You just move from the data polishing stage right into the analysis tool, which can automatically generate your publication-ready Kaplan-Meier curves, log rank tests, and even Cox regression models. It's a true full-circle path from a clinical calculation to a real academic insight.
So it really leaves us with this one question. When you can automate the tedious part of risk stratification and do it in a way that's both instant and completely transparent while also providing a direct pipeline to research, the question is no longer how you're going to do it, but what will you do with all that valuable time you get back?